Hello everyone, my name is Széliová and I'm a PhD student at the University of Natural Resources and Life Sciences in Vienna. And I'm going to talk about why maintenance energy is essential for accurate predictions of intracellular fluxes in CHO. So first, a short introduction about Chinese hamster ovary cells or CHO cells. So these cells are the most popular mammalian cell line for the production of biopharmaceuticals, especially monoclonal antibodies. This is because they are regarded as safe. They can be grown in serum-free, chemically defined media in suspension, and they can perform human-like glycosylation, which is important for the properties of the biopharmaceuticals. However, the cell line development often takes several months, and it is based on trial and error approach. So for example, we need to apply laboratory evolution and then screen thousands and thousands of clones until we find a good producer. And this process is very time consuming and costly and laborious. But now, with the advances in systems biology and metabolic modeling, we could hopefully move to more rational cell line development. So metabolic modeling could help us understand the metabolism of CHO cells better and maybe uncover some metabolic bottlenecks. And this knowledge can then be used to design more rational engineering strategies. And this could lead to faster cell line development. And in 2016, a consensus genome-scale reconstruction of CHO metabolism was published. And this is one of the first steps in successfully applying metabolic modeling to CHO. So far we know that the CHO model is good at predicting growth rates. We saw it also in our data. This is just one example. So here you can see experimental versus predicted growth rate, and the predictions are quite good. And this work was recently published in Metabolic Engineering. And basically we saw that to get good predictions, we need accurate exchange rates, and to get accurate exchange rates, we need to measure the concentrations of metabolites at many time points throughout the batch culture. And then we also saw that it is important to have a cell line specific biomass composition or condition specific, because there was significant variability among the different cell lines and conditions. And if you want to know more about this, you can go see poster of David Rucker-Bauer. So the next question we have is, can the model also predict intracellular fluxes? And this work I'm going to be talking about was inspired by the paper of Isabel Rocha, where they improved predictions of yeast model. And part of this work I did in Portugal at ITQB Institute under the supervision of Isabel. So what we did, we collected literature data about 13C metabolic flux analysis. So there were 6 papers with 20 datasets, and we compared these results with predictions of parsimonious fPBA done with the genome-scale model. So here you can see the results. So these are the predicted versus experimental fluxes, and the different colors indicate the different subsystems. So glycolysis, PPP, TCA cycle, pyruvate metabolism, and amino acid metabolism. And as you can see, a lot of the fluxes are underestimated, and the agreement is not so good. And probably the worst is the TCA cycle, so these blue points, which have very low predicted fluxes, even though the experimental fluxes are really high. So this led us to thinking that there is probably some problem with the energy metabolism. So the first option was that the ATP or NADH are produced somewhere where they shouldn't be. So for example, there could be some wrong reversibility of some reaction, or there could be some cycle which produces ATP or NADH out of nothing. So we checked this, but we didn't find any such reactions, so that was good. So the second option was that there is insufficient energy demand in the model. And the most obvious candidate was maintenance energy. So maintenance energy is a non-growth associated energy consumption, which is used for processes such as maintaining ion gradients, macromolecule turnover and proofreading or for fueling the molecular motors. And so far there is no value available for CHO cells, and there is also very little data for other mammalian cell lines. So I think there is some old paper from the 60s about some mouse cell line, and there are also some estimates for cancer cells, but it's really little. And currently the model contains no value, so it's there is no estimate for CHO cells. So we decided to estimate the maintenance energy, and we did this by scanning a range of different maintenance energy values and then comparing the experimental versus predicted fluxes for each value. And these points here are just different measures of the agreement of experimental versus predicted fluxes, so mean and median relative error, or R². And as you can see, we were able to identify a value where the agreement between experimental and predicted fluxes is the highest. So we did this for all 20 datasets, and we got a range of different values. So on average, they were around 6 millimole per gram droid per hour, but the range is really high. And for example, this dataset is probably an outlier because also the growth rate prediction for this strain was really bad. So it could be that the uptake and secretion rates that they measured are maybe the quality is not so good, or the error is too high. So then we used these mATP values as constraints, and we looked at the overall agreement of the predicted versus experimental fluxes. And as you can see, the predictions improved by a lot. So the R² increased from 0.44 to 0.96. Or if you look at the individual datasets, for all of them, we are able to find some value that improves the predictions. For some of them, it was a very big improvement, and for some of them, just a modest improvement. And if you look at the individual subsystems, the predictions improved for 4 out of 5 subsystems, but not for pentose phosphate pathway. So PPP is predicted to be basically 0. And we thought one of the reasons might be that there is insufficient NADPH demand because maybe some processes are missing in the model. But I have to note here that in a lot of the datasets, the confidence interval for these fluxes were very wide, and in I think more than half of the datasets, it actually includes 0. So we can't really make any conclusions out of this because it seems to be really hard to measure PPP. Okay, so next part of our work is to determine maintenance energy experimentally. So how do we do this? We run continuous fermentations at different growth rates, and at each growth rate we measure exchange rates. And because these are mammalian cells, they grow in a complex medium, and we need to measure a lot of rates. So we need to measure all amino acids, glucose, lactate, and ammonium. Then at each growth rate, we take these rates and we calculate the energy consumption. And then we make linear fit and extrapolate to 0. And this should give us an estimate for the non-growth associated maintenance. So here you can see our experimental results of the exchange rate. So here I have the 4 examples of the most important rates. So this is glucose and glutamine, which are the major energy sources for CHO cells. And as you can see, with increasing growth rate, the consumption of glucose and glutamine increase. And these are the waste products, lactate and ammonium. And here also with the increasing growth rate, the secretion of these products increases. So then we took all of these rates and calculated the maintenance energy. So we did this by feeding all the uptake and secretion rates into the model, and also the growth rate, and maximizing for ATP consumption. And we got these predictions. And the estimate seems to be quite comparable to our computational estimate, but this work is still ongoing. So as you can see, we still have just a few points and the data is quite noisy. So hopefully in a few months this will be finished. And here I would like to just mention some challenges with continuous fermentation of jaw. So it turned out to be very challenging because the growth rates are very low. So it takes a very long time, so like 1 week or 2 weeks to get to the steady state. And also we observed that if the cells are in culture for too long, they start clumping, which makes the cell counting difficult, and also it probably changes metabolism because they cannot mix well if they are in clumps. Then we also had some questionable steady states because in literature there is a rule of thumb that after 5 volume exchanges it should be a steady state, but in some cases we saw that the parameters kept changing, so we had to wait much longer. And another question we don't know the answer to is how low can we go with the dilution rate, because we saw that at lower growth rates the cells start dying, which is a problem. And also it's simply technically difficult because it takes so long. It would take 1 month or even 2 months to do that, and it's just not really feasible to do it in the lab. But we will try to get more points in this area. So this brings me to my summary. So we found out that maintenance energy is very important for the predictions of fluxes by pFBA. We estimated it computationally, and we got a value of around 6 millimole per gram draw weight per hour. And for comparison, these are some values from bacteria, yeast, or mammalian cells that I found in literature. They were either experimental or some computational estimates. And the range of the values is really high, but also our estimates had a big range. So yeah, that's how it is. And the experimental determination is still ongoing. So next we would like to do a 13C flux analysis with our cell line. And I also want to mention a recent paper where they integrated enzyme capacity constraints into the CHO model, and they improved the predictions of fluxes. So it would be interesting to compare or maybe combine these approaches and to see what we get. Maybe our computational estimate would be different, and yeah, maybe the predictions would get even better. So with this, I would like to thank my supervisors, Jürgen, David, and Nicole from Universität Wien, from ASIP and POCU, and also all our colleagues and students. Then the PhD program Biotop, our company partner Novartis, and finally the ITQB Institute and Isabel Rocha for hosting me for the research day. And thank you for your attention.